It's exactly the same. It's exactly the same. Because if you realize sending a man to the moon is easy, it's getting him back that's the problem. And we haven't got the genome sequence. It's stranded on a metaphorical moon. Because we realized at the beginning that the interpretation of the sequence would be the most difficult and the most prolonged effort would be required. And that was clearly the responsibility of science to tackle. So about 18 months ago, I realized that nobody had ever read that sequence. Only computers read the sequence. And as everybody knows, computers are terribly stupid. I mean, they only read what they read, and they can produce, they are fairly obedient, but they can't give you any new ideas. So I thought someone must sit down. So I just sat down and decided to read it. Why not? Oh, it's fascinating. It's unbelievably fascinating. So I started. People said, how did you start? I said, well, started at the left end of chromosome 1. And I read it, you see. And of course, had I been in another culture, I'd have started at the right end, you see. And however, it doesn't matter. Now, what I have to say is that when you ask, what do you learn when you read this? When you read this, you begin to do experiments. Actually, not on humans, but on the genome sequence. So there's a common statement going around which has been iterated many over and over again, is that the human genome sequence duplicated twice. So it's based on the Hox clusters, of which there are 4. And of course, if you duplicate twice, 2 times 2 is 4. Still is 4. And so that's based, and that's done with. And the question is that true? Now, that's a very difficult theory to knock out. Why? Because it gives you an eraser, you see. You duplicate everything, and then to make it fit, you rub out what you don't want. So you have to show, in this case, that rubbing out is contradictory in some way. It is extremely difficult, but that's one of the problems that you can begin to solve from the intrinsic evidence that there is there. Right, so what I discovered in there is that there is an alternative theory which is just based on simple recombination, but with pairing, probably not, but with pairing between transposons. So if you just go through this little hypothesis, and you can work it out quite easily, you can land up with duplicating a little region in a different thing, creating a deletion by the reciprocal thing, and then segregating in one of the many combinations. You can segregate a duplication of some region which has undergone the recombination, and of course the deletion would be lethal, and the other one that will survive is where you have the deletion plus this. So what you've done is moved one batch of genes from one part to another part. Okay? And that says that these events now are the additive. So let me just try and explain this to you in the following thing. I find 2 stretches in the genome, of which there are 5 genes in common, the right sequence with the right orientation. They are they're distant homologs of each other, but the chances that was all independent is 0. Right? But in between them, there are other genes, and they're different in the 2 sets. Okay, so the one theory would say, well, what happened was we had the union of both originally, and then when they separated, when they segregated and developed further, we deleted these three from this one and the other three from the other one. So we landed up, there were originally 6. Okay, that's the eraser. The other theory says, oh no, those 3 just bounced into this position from another place. And since they're not part of a duplication, this means they were just transported into it. Okay? But if you look to see then who has the most fixation events, because that's the important thing, things get fixed in the genome, then in one theory tells you that the one that's deleted has the most steps in it. The other theory says it's the ones that's inserted that have the most steps. So there is a chance to discern the 2 theories by having a measure of steps of fixation. Okay, so that's, I'm just giving you one line of things. But the other line that emerges from it is the rationality of the duplications. Right? And this led me to make another hypothesis, which I will explain and then return. Right, so now what you ask yourself, you go to a meeting on the brain and you listen to people who talk about cell types in the brain. How many cell types do we have in the eye, in the cerebral cortex, in the cerebellum? And I have heard estimates ranging from 5,000 through to 25,000 different cell types. So it's very clear to me that we need a much more rigorous definition of a cell type, because otherwise everything could be a different cell type. Namely, the neuron that has learned something could be considered a different cell type from its next door naive neighbor. So we must have some definition. So the definition is that it's genetically specified. All right? And so this led me to now think of an abstract level in the organism, which I will call decision space. Right? That's the space that says that these are the genes that determine switch points. I make the further assumption that all the switching is binary, all right? And the further assumption that if you lose a switch, okay, you don't become undefined or apoptosed, you simply lapse to you default to the other branch. Okay? Now, right, then we have what I call the Talleyrand hypothesis. Talleyrand was a very famous French statesman who managed to survive everything from the French Revolution through the Reign of Terror, through Napoleon, through two restorations of the monarchy, and came out on top. And he had one dictum, which is, never do for yourself that which others can do for you. Right? And so the idea we wish to pursue is the following. Is there a reduced description in terms of genes for any one organism? And especially, is there a reduced description for humans? Because that's what we're interested in. Now, we say there has to be this because you are not written once. Everybody has an idea that, the genes ought to do, they make a writing, and that's it. You have to keep on making yourself all the time. Okay? And that is every protein in your body is turning over. So to keep you going as a going concern is